Okay, let's start. Welcome everyone to this seminar in systems biology. Really, for me, it's a great pleasure to present Professor Denis Thieffry. He has a very well-recognized researchers in the area of systems biology, especially in modeling Boolean networks, dynamical behavior of regulatory networks, and so on. Professor Thieffry has been working in a variety of institutions, for example, in the Free University of Brussels. He's now a professor in the Institut Curie, in the Escuela Normal Superior in France. And he has been making a lot of publication, almost 115 articles and book chapters in the areas in systems biology. He is currently editor of PLOS Computational Biology, and currently his research is focused on dynamical modeling of the regulatory network controlling in hematopoietic immune cells fate. So for me, it's a great pleasure that you have accepted our invitation, Denis. Thank you so much. The topic that he will talk now is extremely important. Essentially, the problem of how to make reproducibility in terms of science, in terms of computational algorithms, especially using this algorithm, CoLoMoto. He will talk one specific solution to solve this big problem now in systems biology. So thank you so much, Denis, and go ahead.
Thank you, Osvaldo, for this kind introduction. So the idea today is to do two things. Is to give you a bit of an overview of some of our work, but it's also to put it in a more general context. So this issue of reproducibility in computational biology, in particular in the fields of computational modeling of cellular networks. So this is the contents that I am planning to cover. I want to say a few words about this reproducibility crisis in biology, or more specifically in computational biology. And I will focus even more specifically on some assessment of that problem in the field of modeling. Then I will present the solution that we are developing as a community in the field of, let's say, logical Boolean, logical modeling. And I will, for that, also use some reference case studies dealing with T-cell differentiation. I will give you some snapshot of previous studies. I will also mention studies from other groups, but I will particularly focus at the end on one of the most recent studies that we did. And if times allow, maybe I will give you a feeling about how reproducibility is ensured using Jupyter notebooks. And we can have maybe some discussion if some people are online at the end after some outlook. So the reproducibility crisis. This is something that was really stressed about 10 years ago in different fields, not just in biology. And in particular, a publication in 2016 claimed that less than 30% of experimental results published in the journal Nature only can be reproduced by other authors. So less than a third of published experimental results. And in fact, so this was assessed on the sample of studies for sure. And in fact, even worse, only half of these results could be reproduced by the authors themselves. So that's really, so also this problem was already well known. The pervasiveness, the importance, the impact of this problem was not really understood at that level. So the question that we could ask then is to what extent computational studies are affected by this problem of reproducibility? Are they more reproducible? And there is an interesting paper that was published in 2021 by Tiwari et al. That tried to address this question, looking specifically to ordinary differential equation-based models, taking advantage of the fact that there is a centralized repository called BioModel in the European Bioinformatics Institute in UK, where they have already hundreds of models, even thousands of models today. And so they took 455 such ODA models and try to assess the reproducibility of the results that were published in this paper, in these papers, in the corresponding papers. So I'm not going to cover that in very details, but I think it's interesting to convey the main message. And so if it was interactive, I would like to ask the room, what is your guess in terms of success rate? And also a related question, you will see that we have a problem of reproducibility in the field. To what extent this apply to Boolean and logical models, but for that we don't have really quantitative assessment. So this is one of the figures that they published in the paper in 2021, and you see that only about half of the models are considered as directly reproducible, in the sense that you can reproduce at least one of the key figures of the papers associated with the model. And they went a little bit further, because I mean, lack of reproducibility can be caused by can be due to different reasons. Can be just little mistakes, could be some parameters that are not reported, incompleteness in the description, and so on. And at least in some situations, they were able to solve the problem by doing little empirical corrections, and in even less cases, they were able to get feedback from the authors and solve the issues. So, but all together, you end up to less than 50% of reproducible model. So directly reproducible, it is even less, in fact. Then so this is showing how these different categories of reproducibility, let's say, evolve over time, and also with the size of the models. And clearly, bigger are the models, less reproducible they are. So that's quite easy to understand. There are more place for errors. But if we see over the years, there is not so much improvement in terms of the proportions of reproducible papers. So in parallel, there have been a development in computational biology, but probably more related to data analysis. You know, efforts to enforce reproducibility, and there have been also specific articles published to provide rules to enforce reproducibility in computational research. And so here, for example, you have a list of 10 rules, or you could find this number as a bit arbitrary, but 10 rules to help to perform reproducible studies. I will not go over all of them. If you're interested, I think this paper is very interesting to read, but they are quite natural. And they've been also further refined in the case of the use of the so-called Jupyter notebooks. So I will come on that a little bit in a little bit more details later on. But the idea is that you have in one simple document, computational document, commentaries, explanations, and code, and the possibility to execute this code and so visualize the results in one single electronic document. So more recently, so some 4 years ago, a group of people published these rules specifically to improve reproducibility when you are using Jupyter notebooks. And it's not just about putting the right code, but it's also relating to the clarity of the document, the inclusion of explanatory comments, and so on. And then there are, in fact, if you see this list of rules, it goes from very general and relatively simple elements that are applicable, I think, by everybody. And there are also, at the end, some rules that are probably for the more advanced computational biologists. Again, I will not go over all the details, but it's interesting to see that there have been this reflection within the community. In fact, this show the same information is coming from the same paper 4 years ago, where you see that these rules can be organized in 3 groups. One, a series of rules that relates to the organization and documentation of the analysis. Another one that is dealing with the organization of the code itself, to organize the code properly and so on, record all the dependencies, the version, and so on. And then a set of rules that it's more about how to share, design, and explain the document afterwards. And this is a bit of a side, because for sure you cannot get such a well-organized, let's say, electronic notebook in one step. Usually it will require some exploratory analysis, and then refinement, discarding something. Also, it's important to keep track about negative results, maybe not in all the details, but also clearly state what cannot work, and not just positive results. So this slide with this big logo is just to mention that there are a lot of tools and repositories now publicly available, free, where either tools that enable you to generate such electronic notebooks. So I mentioned Jupyter notebooks, which is developed based on the Python language, but that you can also use to integrate codes in other languages. And there is the very popular RStudio and R Markdown system that is more broadly used in data analysis. And then there are repositories to store data sets, to store models, and also to store codes, keeping track of versions, enabling collaborative code developments such as GitHub. Again, I will not go into all the details, but it's important to know that all these resources are now available. They appear during the last decade, and they are getting more and more used. Now, turning to logical modeling, which is one of my main fields of research since my PhD, I was quite struck by the fact that you have so many different software packages. So this list is not exhaustive. I selected packages that are used by various groups usually, and that are still maintained for I think all of them. These packages use different languages, and they are sometimes more complicated to install. they have peculiarities. They are quite complementary. They have complementarities in terms of what they enable to achieve. And so there is clearly a need to be able to go from one tool to another tool to manipulate the same model, and also to compare the results that can be obtained on a given model with different tools, and so on. And this realization led us to form an open consortium called CoLoMoto for Consortium for Logical Models and Tools about 6-7 years ago. In fact, it started even before, sorry, about 10 years ago, but it grew a bit slowly. Now it gathers about between 40 and 50 scientists spread over the world. And it is really, the idea is really to enroll all the people interested in developing tools or sharing tools. And the first thing that we realized is that we needed some kind of standard format to exchange model. That was really the first step on which we focus. And at the time, there was already the systems biology modeling language, SBML, NixML-based format if you want to store ordinary differential equations models, mainly originally for metabolic network that was progressively extended and then revisited in a modular way, which allow us then to define a specific extension for logical models. So this was done within the consortium CoLoMoto with inputs from different groups. It led to a joint article in 2020. It will probably need some revision at some point, but it is now supported by a relatively large collection of tools. So I just show this slide for the people who are from computer science. So you have a well-defined UML diagram. So this is a compact version of it, but where the different entities are organized with their types and so on. And so this kind of format then ensure that once you translate your model, whatever the original format, into this format, then you can also exchange it with people using other software, or yourself use different software. So that was really the first step. And the fact that we add this presentation then enable, so this was the paper, so it's just copy of the first page of the paper, but the fact that we had this format also enabled to share the model through a generic database. So I mentioned biomodels, which was containing mainly ODE based and some stochastic models, since the SBML qual extension, it is now quite straightforward also to submit logical models. Also, there are not so many yet in the biomodels database. So that's it's a first step in achieving some reproducibility. At least the models are stored somewhere. We can find them. We can analyze them with different tools. But this is not enough for sure. What we need also is to be able to keep track about all the models are used. It's not enough to share a model to ensure the reproducibility analysis, but we also need to ensure that people are using the right version of the software, different software, they start with the right initial conditions and combine this as much as possible with explanation about what is at stake, what are the conclusions, and so on. And for that, we again, as a community, we took advantage of the existence of two things. On the one hand, virtual environments which enable to combine and store permanently Linux, in fact, based environment together with a set of tools. And on the other hand, access these tools through Jupyter Notebook. And it's really the combination of these two things that enable really good reproducibility of the analysis of logical models. So a few words about CoLoMoto. It's evolving quite fast. This is the slides coming from the seminal papers that we publish. At that time, we had like 6 or 7 different tools, including CoLoMoto, but now we have many more. In fact, there are new tools appearing every month or so in the collections. Especially that also Python is getting more and more used as to develop software tools, and they are easily integrated for sure in this system. But we have also other tools, C++ tools and so on, that are integrated in the virtual machines implemented with Docker. And thanks to, in particular, the standard format for the models, but also some conversion functionalities, it's possible to go from one tool to the other tool very easily with one command line using the CoLoMoto notebooks. So here we have this subset of tools that were available 5 years ago. So the environment really is access to different tools to compute various interesting properties of logical models. And it also enables, because of this notebook part, the Jupyter notebook, it also enables to document the analysis workflows and provide in a simple, in a single object document the code, the results, and the comments. And this can be, for example, proposed as supplementary material to an article. Now, I will mention very briefly that there have been additional efforts made by the CoLoMoto consortium, in particular to define the best practices and standards for the curation and the annotation of logical models. So this article was published following a workshop specifically dedicated to these questions some years ago. And now I want to turn to a specific application. I want to give a bit of a historical perspective to it, just to indicate a bit how the practice have evolved. And I think what is the current state of the art to propose and develop models. So referring here to T-cell differentiation. So to my knowledge, some of the very early models for regulatory T cells and interaction between different types of human cells was in fact the work of Marceline Kaufmann. This is not Stuart Kaufmann. Now about almost 40 years ago. The concept have changed. At the time they was speaking about T helper cells, but also T suppressive cells. These notions have been evolving since then, and she proposed a very small model with 4 variables and covering these different cell types. So it was very phenomenological, and then there have been a series of publication over the years. So this is just a partial list for sure. I wanted to mention some of the contribution from Mexican group, in particular, Luis Mendoza has been very early in proposing molecular networks controlling models of molecular network controlling TL per cell differentiation back in the early 2000s. And then my group proposed several models that I will mention now briefly. But before that, I want to point out that initially this model were mainly presented and shared through textual description and graphs or graphics, sometimes tables, supplementary tables with rules and so on. And then over the years, we start to share model files and software. Initially, people were just using their own code and integrating equation without mentioning very precisely how they were doing this computation. And then at some point, people start to also to share, in particular my group start to share the latest models, not only the model themselves, but also the full analysis workflows as a notebook and a companion Docker container. So I will go quickly over a few snapshots for former studies, and then I will spend a little bit more time on the very last one. And in this process, what I would like to strike is that models are usually developed to address specific questions, but they can be reused, modified, extended, sometimes simplified to address new questions. And so sharing of models and analysis workflow are also useful to enable further studies, not just reproducibility. So the problem is here explained in a cartoonish way. We are interested in the differentiation of these naive TRP cells, which play a very important role in the control, the modulation of the human response towards pathogen or cancer or different treats. And we know that they can differentiate in different subtypes, and this will depend, on the one hand, on signaling that is received by these naive T helper cells through various signaling pathways. I will mention a few more later on, and then the specific expression of some transcription factor and also of some specific interleukins. So signaling molecules that will then affect other cells. So that's how they control the immune response. And depending on which transcription factor and interleukins, we speak about Th1 or Th2 or Treg or Th17 subtypes. There are more of these subtypes, but I want here to illustrate the concept using these 4 subtypes. For example, Th1 is very important for what is called the cellular response, so a response that will involve, for example, cells that can specifically kill or phagocytes foreigners, invaders, or there are T helper cells called Th2 that will trigger the production of antibodies by B cells. There are specific T helper cells that can damp the immune response, so suppress the immune response, and thereby avoid, for example, inflammation. And there are, at the opposite, T helper cells, in particular the Th17, that are more prone to trigger inflammation. And there are more subtypes, each with their characteristics. So I mentioned the seminal work of Luis Mendoza. I will not go over his own contribution. He was looking mainly at Th1, Th2 differentiation. But in 2010, we proposed a more comprehensive model that took into account about 10 different interleukines that can affect this differentiation process, different transcription factors. And I will not go into all the details, but I just want to mention the key finding, which was that this model, depending on the combinations of input and so on, can give rise to a quite large number of different stable signatures in terms of expression of interleukines and key transcription factors. So in this matrix, what you see is that each row will correspond to a given stable state, and each column corresponds to one of the components of the network. They're not all there. They are, I represented only those that enable to identify the TL per cell subtypes. And what you see is that just by looking at the name, we tend to have several stable states corresponding to one TL per subtypes. So that's a first refinement. And all these subtypes here at the top can be considered as canonical subtypes because they express one key transcription factor associated with one of these well-known subtypes. But we, at the same time, we realized that with this model, there was no way to avoid to generate also to hybrid subtypes. And at the beginning, we were very much unknown. We thought, okay, there is something wrong in the model. And then we discovered that, in fact, this really exists. There were some reports for most of these hybrid subtypes. So here, the modeling really helped us to understand that what we call a cell types can be a bit more complicated than just a stable state. And there is clearly some plasticity and some possibility to have sometimes co-expression of markers belonging to different subtypes. And in fact, exactly the same year, a very influential review by two key players in the field published this cartoon in a review where they not only clearly mention the existence, for example, of a hybrid subtype here co-expressing two transcription factors that were considering as mutually exclusive, but that you have also possibility to reprogram one TL per cell type into another one without really specifying what is necessary for this reprogramming. These were like more like observations gathered from different articles. And so this trigger for, so yeah, they mentioned hybrid subtypes and reprogramming. And this trigger from us the idea to go further and systematically analyze the possibility to reprogram cells in silico. So the first thing we did is in collaboration with an experimental group, in particular Maximilien Grand-Clodon in the group of Vassilis Humalis in Paris. And so this was the work of a postdoc in my group called Wassim Aboujaoudeh. We further extended the model to take into account more cytokines that they could use in vitro in the lab. And again, I will not go into all the details, but the idea is that you have all the cytokines at the top here, and then transcription factor with many cross-regulation, inhibition in red, activation in green, and then some cytokines that can be expressed directly by the T cells, depending on its regulatory states. And so this model is getting really more tough to analyze. And in fact, this development led us to, on the one hand, to develop new methods to cope with the large size of these models, in particular model reduction methods. On the other hand, also to try to be very systematic to record all the analysis steps. Not yet with notebooks, but going in that direction. And I just want to show one key results from that paper. In fact, we use for that also tools coming from the computer science community called model checking tools, in particular new SMV, to check to what extent by changing the microenvironment. So this is correspond to the labels on the arrows. It corresponds to a change in terms of cytokines in the environment. So we wanted to check to what extent we can go from a stable state, for example, corresponding to H1, to a stable state corresponding to TH17. And indeed, if you put the proper, the known cocktail, which enable to differentiate normally TH0 in TH17, you can get the reprogramming of TH1 on TH17. And so you see that now here, this graph represents different possibilities of reprogramming for very specific conditions that are labeled with this pro TH2 and so on things. And you see that it is it's not symmetrical at all. Some nodes receive a lot of arcs, so it means that it's quite easy to reprogram subtypes into that subtype. But some others, so it is the case for TH1 and TH2 that are well-established TL per subtypes, but others seems to be much more labile, like TH9, where you have one arrows enabling the differentiation of naive T cells into TH9, but you can very easily, reprogram these cell types into other cell types. So let me explain also why for example, TH9 subtypes was discovered much later than TH1 and TH2. It's more labile, more difficult to capture. So time is flying, so I will try to be short for the rest of the talk. I want to mention that based on this model, many extension has been done, in particular in Mexico, in the lab of Maria Angelica Santana. So this study extended, in fact, or adapted our model and also reduced it to some extent, but to add interactions with an important toll-like receptor pathway involved in T cell activation, in particular neonate T cell activation. Another PhD students from Angelica look at the role of metabolic status and contribution of ROS spaces in the activation of T cells, in particular in neonate T cells. So I will not cover that, but you see that you can reuse the model, readapt it, maybe leave some part, add some part to address new questions. That paper comes from my group, but I will not cover it also in details. But in that case, we want to expand the model to take into account the role of immune checkpoints. And we end up with a model very large, and this also led us to make new methodological development. But the thing I wanted to present in a little bit more detail was the following problem. Up to now, we were, let's say, following a bit the general trend, which is to try to account for the different subtypes that were clearly identified by immunologists. But with this study that I'm presenting now, in the lab of Vasilis Soumelis, the idea was to make a more systematic analysis, try to start with a lot of different initial conditions, playing with different ways to activate dendritic cells, in fact, and presentation of antigens by dendritic cells to T cells, measure as many signals as possible from dendritic cells onto TL per cells, and then measure as many signals as possible, in particular using FACS, cytokines at the level of T cells. So the idea was really to to get an idea about the diversity of TL per cell profiles, gene expression profiles, by maximizing the diversity of signals towards TL per cell. And so we could gather a lot of information. So this is a set of gene expression values using, I think in that case, it was PCR. We also got transcriptomic data at that time using DNA arrays, gravimetric DNA arrays. And we also got information about specific cytokines using FACS, as I mentioned. And one thing that really strikes us is that we realize that what we call up to now the signature of Th17 subtypes was the expression of the IL-17 proteins, but lumping together, in fact, two different proteins called IL-17A and IL-17F that are encoded by neighboring genes in the genome. And what we realized at that time is that when we put the cells in conditions where we are supposed to get Th17, we do get expression of these two cytokines, but usually at the single cell level, you see that most cells tend to express, in fact, only few cells express both IL-17A and IL-17F, and most cells that will express only one of them or none of them. Okay, so this led us to try to address this question using our model. But our model was lacking details. We needed to be able to distinguish between the regulation of these two cytokines. And we had some evidence that a specific cocktail of cytokines in the culture medium could trigger Th1 or Th1 Th17 maybe response, or Th17 response. That was our initial setting. But later on, we realized that we needed also to account for the independent expression of IL-17A and IL-17F, which may have important clinical impact because they are involved in different, for example, autoimmune disease and so on. So starting from the model that I presented you before, we select some signaling pathway, and then we try to extract information from recent high throughput data, in particular ChIP-seq data, to implement regulatory mechanism for the regulation of IL-17F and IL-17A. So and then because this was quite still hypothetical, also there is some molecular evidence, we could try to assess to what extent the resulting model could account for observation. And so the first thing we did is to see that the model can reproduce the canonical subtypes that we had before, Th0, Th17, and Th1. So here in red, it represents active component, and green inactive component. So you see that when you have IL-17 expressed, you have both expressed, like in the classical situation. What we thought before was happening. So this may still happen, but we can now also account for situation where we have only one of the two expressed, and this led us to define more subtypes depending on this expression of one or the other or the two IL-17. And in the next step, what we did is to perturb the system. So just blocking the expression of one key transcription factor upstream of these cytokines, and with the aim to check then what happens in terms of expression of the IL-17 A and F for different combinations of inputs. The one that were protease 17 and the one that was initially enabling the expression only of IL-17F and not of IL-17A. And by doing that, we end up by putting the finger on key transcription factor involved in this different differential expression. And we could also use additional tools to check to what extent, because in simulation, it happens that you, from a given initial states, you can reach several stable states, and adding a stochastic layer to our simulation, taking into account up and down rates, in fact, equiprobable up and down rates, it is then possible to obtain this kind of pie chart. So telling you that in some conditions, if you start from a naive initial conditions, you will get mostly cells expressing IL-2 or other cells that do not have this profile at the end of the simulation, or you can reach different profiles, and including profiles that would correspond, for example, to the expression of only IL-17A, and so on. Now, I want just to wrap up the conclusion for this study. This led us to propose a specific molecular mechanism accounting for the different expression of the two interleukin 17. As I mentioned, they are located on the same chromosome at some distance from each other. Each of them are controlled by promoter region. And for the IL-17A, there is also documented enhancers. And what we realize is that for some specific input conditions, so in the presence of IL-12, sorry, and IL-1 beta, you get some autocrine activation of the IL-2 pathway, and this in fact impede the expression at the end of IL-17A, sorry, whereas the expression of IL-17F is enabled. In the classical protease 17 condition, you can get, in fact, both IL-17 express, because in fact there is no interference with the activity of the enhancers and the promoter controlling IL-17A. And finally, in other protease 17 condition, you may end up with a situation where you have expression only of IL-17A, but not of IL-17F. So that will depend really on the dynamics and what happens along the way. But this led us, because we have this mechanism, then to identify key transcription factor, in particular STAT factor or CN or R, as playing a role in this decision to express one or the other or both IL-17 cytokines. And in a snapshot, we were able to confirm the fact that the expression of these two cytokines can be uncoupled. We could point to key transcription factor involved in their differential expression, so NFAT2A, STAT5A, and SMAT2, namely. We could also show that with the model and then experimentally, that the production of IL-17A correlate with an increase of SMAD2 transcription, whereas the expression of IL-17F correlate, in fact, with an increase of BLIMP1 and STAT5. So we have different transcription factor controlling or playing a key role, let's say, for the regulation of the expression of these two genes. And as a conclusion, we could propose the first mechanistic model accounting for this differential expression that was suspected but not clearly established before the systematic experimental work from the group of Vasily. And this could be important, in fact, to design novel therapeutic intervention, in particular for autoimmune and inflammatory disease, because these two interleukins have different effects and can be deleterious depending on the disease to be cured. And finally, the CoLoMoto environment enable the reproducibility, the reusability, and the adaptation, the extension of this model. In fact, we use the CoLoMoto environment to take the preceding model developed by Abuja et al. To simplify it and then make this analysis. So I'm done. I just wanted to show you how a notebook look like. If you have just 30 more seconds, but I will not go through it, so it is here. So it's a brute notebook here, but you see you have command lines, and we can add text and so on. In that case, we didn't put much text. There are some comments, but and then you have all the computations together with the text. And this is used together with the virtual machine that I mentioned with, so using the Docker system. It's also possible to use other virtual environment, but right now we mainly use Docker to combine the logical modeling tool. So I'm ready to take some question. If there is some people in the audience, there is something in the chat. Ah, ok. Bienvenidas sus preguntas para el Dr. Jeffrey en español también. Yeah.
Okay. Sí. Okay. Thank you so much, Denise, for this gorgeous lecture. I have some questions. Meanwhile, some other write their question in the chat. My first question is, really, this is an important part to try to make reproducibility, for instance, in terms of the modeling. In the area of databases, there is another type of philosophy, a current philosophy, from my point of view, that is called FAIR. That is yes. The initials of findable, accessible, interoperable, and reusable, the database. Do you think that this can be used here in order to look for more ambitious, maybe objectives, for instance, to try to connect many reconstruction of immunological cells in order to make a bigger behavior and analyze the phenotype behavior for more components? What do you think?
So what I presented with CoLoMoto is really guided by this FAIR principle. Okay. Maybe we didn't have them in mind at the beginning, but they are quite natural. Huh? So it's nice that they've been, stated clearly at some point, but we had really this principle in mind, and it's not directly related to the size of the model. A small model can be very compliant with FAIR principle. I think it's really it requires to pay attention to a series of things, and I think that current technologies, those that I mentioned at the beginning, really help us to do it. And it's even not just because FAIR suggests, the English term means also to be fair towards others, right? But right. And so it costs an effort. But in fact, FAIR is also very beneficial for the person, and in particular for the group where the person sits, because it helps to keep track of all the work done and all the information at some point, used to, for example, to develop a model. So there is I didn't really went into the details, but you also need to annotate your model in details, and so this is not done in the notebook. It's done at the level of the model itself. In GeneSim, you can annotate it, attach references and so on, and in other software, it will be maybe put as comment, or so you can see a model as like a mini database already. With a lot of references, links to external database, and so on. So that's how we develop our models. And then there was one side at your question that sounds like we should try to make everything interoperable, meaning that maybe by having many models that can talk or that can be connected together, we could have like the grand picture, right?
Right.
This is, I'm a bit more doubtful in the sense that for me, a model is really developed to address specific questions. It doesn't mean that it cannot be reused to address other questions, but usually it has a cost. You have to revisit the model, add stuff, delete stuff. That's what I was trying to illustrate with the different steps or the different studies that I mentioned a bit quickly. But it was not just extension. Sometimes it was saying, oh, okay, for that question I don't need that, and I take out stuff. Because it would not be helpful to keep this stuff. And so you focus on what is important for your question. So I think what is important is that you can share things and justify really well why you put this and this, but not necessarily that everything should be piece of a puzzle that would make sense as a whole, as a single big thing. So that's my feeling, yeah. But
I see. Me, on the other hand, meanwhile, I was hearing this paper that you published about the 10 simple rules for the reproducible computational research. I was very aware about the criteria to include. For instance, taking into account the documentation of the process, the building of the pipeline, using tools like GitHub, Git, Docker, Jupyter, like you show at the end. However, in one point I was thinking about when you write share your data. Of course, share the data is extremely important in order to reproduce. However, if the data are in the clinical area, for instance, maybe measurements about something that can be more protected by regulation, I don't know. How try to balance this type of to share the data by one hand. However, you have the opposite component, no? The privacy of this data. What do you think? Maybe in these models are not very, is not the case, but in other cases, like the analysis of clinical anthropometric measurements or things like that, there are a privacy component. What do you think about it?
So there are really, again, two parts here. One is privacy because it is touching to the integrity of a person. So, for example, the genomic sequence of somebody should remain private, or at least not. I mean, the person should not be identifiable. And it's important because otherwise it can lead to misuse by insurance company or whatever. They will, try to compute prognosis of, what is the probability you get cancer and stuff like that. And that would be very unfair. And it probably be also wrong because our way, for example, to predict that a person will have cancer is, I mean, it works statistically, but at the individual level, you cannot say. Maybe you have 30 chance to develop I don't know, a lung cancer if you are smoking, but you may perfectly not develop a lung cancer. I don't know. So this is one thing. I think we need to keep some privacy or anonymous, you know,
Exactly.
keep anonymous some information. But the other thing is more, you know, the money aspect, so patenting and so on. And here my position, but again, it's a personal position, is that it's a position for open science. I think we do science with public money, okay? We produce information based on information of other person, in particular in the field of computational biology. And I think you can patent, let's say, a mechanism or something, or a solution to a problem. But patenting just a prediction or information that you produce with algorithm with public data, I don't feel at ease for that. And I think I mean, there are companies that try to do it. It's a bit like more treating than really legal enforcement. But so myself, I always share, and now I think, for example, there is a new practice, and I think it's really good, is first to publish preprint, and I think it's beneficial for the authors because usually you get interesting feedbacks when you publish a preprint. And it's proof that you have, you know, achieved some results before you have finished the more and more complicated process of peer reviewing and so on. And the other thing is that there is also a tendency now to favor open access journal, and I think it's particularly important for countries where you don't have all the resources to pay for all the to access all the journals.
Okay, okay. Thank you so much.
There is a question in the chat that I think is interesting. So the question is about cyclic attractors. So what is their biological interpretation? If they would imply some reversibility in the polarization of cellular states. So there is a tricky point about a cyclic attractor in the field of Boolean networks. You probably, the person who is asking the question probably knows the difference between asynchronous and synchronous updating. For a long time, people were using what is called synchronous updating. So using the logical rules, changing the values of all nodes of a network at the same time, depending on the rules, naturally. And then you have a unique successor states for each state. So you can have convergence sometimes, and sometimes you end up in a cycle, but there is no possibility of differentiation in the sense starting from one state and reaching two different stable states, for example, or you need a perturbation to achieve a change of trajectories like that. So I think this is a bit an artifact, an oversimplification. But the problem is that when you do asynchronous simulation, you get much more complex dynamics. And in fact, it's more like an over-approximation of the real trajectories. And so this has been quite confusing for many people. And it's also more complicated to compute. Now, with the computer that we have, we can really make sense of asynchronous updating. And there are more and more people using asynchronous updating, even for large networks. So when we speak about cyclic attractors, we have to be clear if it's something that is really robust. It's not only an artifact of synchronous updating, because this may happen. But you have also well-defined cyclic attractors that are robust using whatever synchronous or asynchronous updating. So if we have a bona fide cyclic attractor, a robust one, I think it does usually represent something from a biologic point of view. It can be a cyclic behavior, something really oscillating over time, or it could be sometimes just the fact that you tend to, like a thermostat, you tend to keep your variable value around some optimal that is really between 0 and 1, so along the threshold, if you want. So you have really two different interpretations. Often the small cycle, like 1 or 2 component cycles, will tend to be more of the thermostat-like, whereas a larger cycle involving several variables will represent real oscillatory behavior. And there are two very good examples in biology. You have the models that enable to account for cell cycle, and for sure you need a cycle representing the sequence of phases. And it's I mean, what is interesting about the question of reversibility, in fact, each transition is reversible, but at the end you make a cycle. So you don't go backwards in the cell cycle, but you end up back to the G1 state at some point. And the other example is circadian rhythm. So we have also good models with both DEs and logical framework to account for this cyclic behavior. So I don't know if I answer completely to the question, but
yeah, I think so. In the same direction, I have I would like to say that in your models you make asynchronic and synchronic simulation, and do you remain, this attractor remains the same, or
so the stable state are the same. The stable state are conserved. There is no transition, so they are common, whatever the updating strategy. There is a part of the cycles that are conserved. The cycles where you have only one transition in
part
each
of
state.
the cycle, only part.
If you have a sequence of states such that you have only one node changing, you know, a call to change at each step of the cycle, then this is conserved also. And even more profound, there is a concept that is getting more and more used in the field, which is the concept of trap spaces. There are different ways to compute them. And these are set of states such that in fact, there are hyperboxes in the Boolean space. And once you are in this hyperbox, you cannot go out. So it's an approximation of attractors. So stable state are special cases. They are just one-dimensional, you know, one variable to one. Sorry, it's a point attractor or one cell in the Boolean space attractor, and so it's also a trap space, because when you are here, you cannot go out. But a cyclic attractors will be like involving some states within a box, and they can be approximated by a box where you tend to go there and you cannot go out. Okay. And these are it depends exactly how you define them, but there is a good way to define them that is invariant, does not depend on the synchronous or asynchronous updating. So trap space are conserved, but what is in a trap space may change depending on the synchronous or asynchronous. So some kind of approximation, you know that if you are in that part of the phase space, you are trapped there. So this will be true whatever synchronous or asynchronous updating you use, but what is happening exactly within this box will differ.
I see. Okay. Oh, you answered the question of Aaron. Great, thanks.
Yeah.
He said.
Yeah.
Okay, perfect. My last question is in terms of the stochastic Boolean models. Denis, when you try to reproduce, of course, there is a noise inherited in this type of dynamics. In that case, what is the idea to report to make reproducible? Of course, the average, the moments of the distribution for the levels of the expression, or what is the most proper way to say it? This is the main result, and if you reproduce, even considering the stochasticity in the environment, you will get this data, the same data.
So indeed, when you maybe I can show you. Well, I didn't show time plots, but I showed a pie chart, right, with percentage.
Yes, yeah.
So the exact percentage may differ if you run exactly the same notebook twice.
Exactly.
The exact percentage may differ, but it will be really very marginal. You can, if you want to be sure, because it is important, but usually for us, it's not very important, the 0.1 or 0.01 difference in the percentage. But if you want to ensure that you get exactly the same percentage, then you need to encode a seed for the random choice. So it is possible to do. In fact, in some studies, it is important at the same time to do a stochastic simulation, but keep track that you have initiate the computation with a seed for the computation of the random numbers, because it may, in some cases, it may affect the results in a significant way. For all studies, it doesn't matter. You will see almost no difference in time, because we also perform simulation a lot of time, right? We will do 1,000 simulation, and what we show are means. So except for very exceptional results, we don't expect to see
I see.
Significant variations. Yeah.
Okay. In terms of CoLoMoto, I was about some years ago. I had the opportunity to know René Thomas and Marceli Kaufmann, great persons and great scientists. And my question is, René Thomas has been developed this discrete level of dynamical behavior for the cells. CoLoMoto include this type, or in the future?
Sure.
Okay,
so, perfect. GenSim, in fact, account for. I didn't insist on that, but if I go back to the slide, I don't know where there. They are hidden here. Sorry. In our model are largely Boolean, but not completely. This model has a few, the square nodes here can take 3 values. And there is some justification for that, biological justification. It's because interleukin-2 a receptor can be made with different chains, and some have higher affinity than others. And so you have a stronger signal. And in fact, the referee of our paper asked us at some point to take that into account. So we did it, and our formal is unable to do it. Within the CoLoMoto package, you have tools that enable the use of multi-level variables and other not. But there is you can always re-encode a multi-level model into a Boolean one. There are a few things to be careful about. So you should not try to start from states that makes no sense in the translated Boolean models. So typically see if you have a variable that can take 3 values, you will encode it by 2 variables that can take 2 values. One will tell you I am above the first threshold, and the other will tell you I am above the second threshold. And so it would make no sense to say that the second variable set the value 1, but the first one has the value 0, because you have been above the second threshold, but you are not above the first one. So there are some tricks to take care of, but you can translate a multi-level model into a Boolean one. And Mabos, for example, works only with Boolean models, but we can perfectly translate our multi-level models into Boolean models that you can then manipulate with Mabos.
I see.
And so great. This is done automatically, in fact. There is a function from GenSim that export that Boolean as a model. Yeah.
Okay. So if there is no more questions, I would like to say thank you again, Denise, for this.
Thank you for the invitation.
Thanks so much. And we will see soon to talk more about this type of modeling.
Okay.
Thank you so much. Thank you,
So,
everybody.
Goodbye. Have a
Bye.
good day. For me, it's the evening, but
Evening,
bye-bye.
okay. Buenos días.